Good day. This is to present to you this study entitled Isolation, Purification, and Molecular Identification of Microfungi from the Landfill site of Bangar, La Union, Philippines. The researchers are yours truly, Mark Kevin S. Lopez and Francis Ruel G. Castillo. For the rationale of this study, fungi are an extremely versatile class of organisms which are comprised mostly of saprophytes, living on dead organic material and are relatively diverse in terms of forms and the environment they live in. Of the 1.5 million species of fungi, only 10% of which is known and named. Hence, it is then very important to describe, isolate, and identify fungal species of great potential which might not yet known to science. This study generally aimed to establish the baseline information for soil-borne microfungi present in the landfill site of Bangar, La Union, Philippines. Likewise, it is specifically sought to 1. Evaluate the pH, organic matter, and heavy metal contents of soil samples from the landfill site. 2. Isolate, purify, and describe the microfungal species present in the landfill site. And 3. Identify molecularly the purified microfungal species isolated from the landfill site, and assess the phylogenetic relationships of the different microfungal isolates. The methodology include collection of soil samples, soil analysis, preparation of culture medium, isolation and purification of microfungal isolates, description, identification, and phylogenetic analysis of microfungal isolates. In collection of soil samples, systematic spatial grid sampling technique was employed, wherein soil auger was used to collect around 100,000 grams of dry sample from a depth of 50 centimeters in every spatial grid sampling location point. The collected samples were then mixed in order to come up with a 9,000 grams composite soil sample. The collected soil samples were placed in bags and brought to the laboratory. In soil analysis, soil sample was sent to the CRL Environmental Corporation Environmental Testing Laboratory in Pampanga, Philippines. The soil sample was analyzed in terms of pH, organic matter, and heavy metal contents. In the preparation of culture medium, commercial potato dextrose agar was used as the solid culture medium, which were prepared and placed in earthenware flasks sterilized at 100 degrees Celsius and 121 psi for 30 minutes. The sterilized PDA medium was poured in Petri plates and allowed to solidify. In isolation and purification of microfungal isolates, about 0.02 grams of pulverized dry soil were seeded in the Petri plate with PDA medium, and it was sealed with parafilm. The plated samples were incubated at ambient room temperature, allowing growth of fungal colonies within 3 to 5 days of incubation. Distinct fungal colonies were isolated by individually picking them using a sterile inoculating needle and transferred aseptically into Petri plates with PDA. Three-point inoculation technique was employed to assess the purity of the culture, and once pure culture was obtained, it was inoculated in bottles with media, which served as the stock cultures for fungal isolates. In the description, identification, and phylogenetic analysis of microfungal isolates, the illustrated manual on identification of some seed-borne Aspergilli, Casaria penicilli, and their mycotoxin by Sind et al. 1991 was used. In the description and identification, by observing the macromorphological characteristics like color and texture of the obverse side of the colony, and micromorphological characters like types of hypha, color of mycelium, structures of their asexual spores, and presence of other structures like apophyses, chlamydospores, and sclerotia of the microfungal isolates. In molecular identification, The microfungal samples in tubes were sent to the Macrogen Incorporated in Korea for the DNA extraction, PCR amplification, and sequence analysis of the different isolates. The molecular identification were performed in order to confirm the identity of the different microfungal species. The primers ITS1, which is the forward, and ITS4, which is the reverse one, was used to amplify the ITS region, and the amplified nrDNA ITS sequence was used for blast analysis to determine the identity of the microfungal species with the highest percent similarity match in the NCBI database. Phylogenetic analysis of the different isolates was also performed using Mega11 software with the statistical method as maximum livelihood, test of phylogeny as bootstrap method with 1000 replications, and substitution model as Kimura-2 parameter correction method. The results include soil analysis, the microfungal isolates, and molecular identification and phylogenetic analysis. In soil analysis, we have here Table 1 showing the pH level and organic matter content of soil sample from the landfill site of Bangar, La Union, Philippines. The pH is slightly acidic to neutral, which is 6.5, while the organic matter content is 3.07%. And according to literatures, the organic matter, which is 3.07%, is comparable to the organic matter present in agricultural lands. On the other hand, Table 2 shows the heavy metal content of the soil sample from the landfill site of Bangar, La Union, Philippines. 9 heavy metals were analyzed, 3 of which were not detected, namely arsenic, lead, and mercury, most of which are within the permissible limits set by the World Health Organization except for cadmium, chromium, and nickel. The microfungal isolates. In this study, 10 microfungal species were isolated and purified belonging to 4 genera such as the Aspergillus, the Penicillium, the Acrophyalophora, and the Actinomycor. 10 microfungal isolates were described and identified, 7 of which were confirmed through molecular technique. This figure 1, shows the 1-point and 3-point cultures on PDA of the different isolates after 7 days of incubation, as well as their microscopic conidiophores. A is Aspergillus oryzae, B is A flavus, C is A therius, D is A candidus, E is A fumigatus, F is A niger, G is Penicillium oxalicum, H is Penicillium species, I is Actinomycor species, J is Acrophylophora levis. Table 3 shows the morphological characters of the previously mentioned microfungi isolated from the landfill site of Bangar, La Union, Philippines. The different microfungal species were characterized in terms of their overse colony, their color and the texture of their overse colony, their sporangiospores, conidiophores, and as well as sporangiospores and conidia. In molecular identification and phylogenetic analysis, Table 4 shows the species list including isolate numbers, genbank accession numbers, PP length, and percent similarity used in this study. The genbank accession number, particularly the ITS region, is presented for reference. We can observe that the BP length of the different microfungal species ranges from 571 to 605. This is within the range of the BP length of the ITS region, which is 600 to 900 BP. And then figure 2 shows the phylogenetic tree Based on maximum livelihood analysis of ITS sequences of the 7 microfungal species, bootstrap support values of greater than or equal to 50% are indicated at the nodes, and the bar indicates the number of substitutions per position. We can observe in the figure that the Aspergilli species were closely related to each other and distantly related to Penicillium and Acrophylophora levis. For the conclusion of this study, the pH, organic matter, and heavy metal contents of the soil from the landfill site Bangar La Union is within the permissible limits, except for cadmium, chromium, and nickel. 10 microfungal species are isolated, purified, and described, namely Acrophylophora levis, actinomycore species, Aspergillus candidus, Aspergillus flavus, Aspergillus fumigatus, Aspergillus oryzae, Aspergillus tyrius, Aspergillus niger, Penicillium oxalicum, and Penicillium species. Phylogenetic analysis reveals the relationship of the different microfungal species isolated from the landfill site. For the recommendations, the study recommends that the heavy metal contents of the soil of the landfill site must be periodically monitored in order to prevent ill effects to the environment and the people. Further studies like heavy metal uptake or tolerance can be made on the microfungal isolates to determine potential microremediation agents. Studies on growth-promoting effect of microfungal isolates on plants can also be explored. Thank you.